Hello, everyone. Welcome to the second webinar for 2018 on collaborative modeling of the brain. I'm Matteo Cantarelli. I am the CTO of MetaCell. I'm talking to you from Oxford in the United Kingdom. And first thing, I want to thank you to taking the time to join us today. And I am really pleased, we're all very pleased to have with us here Dr. Porrick Gleason with us from the Open Source Brain Project. Hello, Porrick. How are you?
Hello. Good, thank you.
So Porrick is one of the founders of the Open Source Brain Project, which started in 2012. He is a senior researcher at the University College London, where he has worked on a number of computational resources, including NeuroML, a standard that many of you probably knows for modeling computational neuroscience, and NeuroConstruct. So a little bit about our company. As always, MetaCell is 6 years old, and at MetaCell we're passionate about building software for neuroscience, a lot of which, as you've seen throughout this webinar series, has a lot to do with visualizing and sharing data and models online to help researchers and teams collaborate together. So as always, throughout the webinar, we're going to have some interactive polls where we'll be asking you some questions to get a better sense really of our audience and what your interests are. And you will be able to ask questions throughout the webinar using the interface on your screen, and we will answer the questions at the end of the presentation. So as MetaCell, we work with a large variety of organizations, which span from universities to pharmas, small companies, and non-profit. So first thing I would like to do is launch a quick poll to see a little bit what is the breakdown of the room, which should be coming up on your screen now. So this is just a way for us to understand whether you work in academia, industry, or other. So so far we have 74% of the vote, which is quickly going up. Thank you for everyone that is taking the time to do this. Okay, there are a few more. All right. So breakdown for today is, you should see it here. So we have 50% from academia, and the rest split between industry and others. Okay, so what will we talk today about? So we'll talk about Open Source Brain. Open Source Brain, as you'll see, is a platform for visualizing, simulating, disseminating, and essentially collaboratively developing standardized models of neurons and circuits from a range of brain regions. Open Source Brain is entirely web-based, and so it's something that you can easily just use essentially with your browser without needing to install any additional software or simulator. Which is makes for a great resource, really. So you will see, and we'll see with Porrick shortly how that works out. Open Source Brain is developed at the University College of London in the lab of Professor Angus Silver, and is funded by the Wellcome Trust. So as MetaCell, we've helped building Open Source Brain. In particular, we helped build a Geppetto-based application that is at the heart of OSB, and it essentially enables the system to load the NeuroML files, to visualize them, and simulate them using the neuron simulator on the backend. So I would like now to start asking a series of questions to Porrick, that has been gracefully given us his time to share with us Open Source Brain here. And I would like to first to take a step back and Porrick, could you describe what a computational model is? Taking a step back, what a computational model is for those that are listening in and are not familiar, and also maybe if you could say a little bit about what are the different approaches that exist when we're talking about modeling.
Okay, great. I'll happily give a quick introduction. Let me see, can you see my screen there? Yes.
We are. Yes, thanks.
Okay, great. Okay, so computational modeling in neuroscience, in a similar way to all areas of neuroscience, looks at various different levels in the brain to try to understand how it functions, how information is processed. So we know that information is processed in at the molecular level, at the genetic level, but also within different areas of brain cells. So, for example, at the synapse, there's quite complex interactions, individual neurons and networks, and obviously at the whole brain scale. So, experimentalists are studying the brain at all of these different levels, trying to figure out what's going on. And in a similar way, computational models are created at all of these different levels. So, on the right here, you'll see that at the bottom there's some examples of gap junctions, some examples of synapses. In the middle, you see individual cell models being modeled as electrical compartments. And at the top, there's a model, a 3D model of a network, a cerebellum. And all of these different types of models are being created. There have been publications for the last 20, 30 years creating these type of models, and this is really what's motivated open source brain to try to get a greater amount of sharing of these types of models and get much more accessibility for modeling in neuroscience as a whole. So if I just click on here, so just to say a little bit more about two different types of approaches that have been taken to modeling networks, for example, in neuroscience. At the top, you have one approach which looks at the individual neurons and takes a very simplistic view of what goes on at the individual neuron level. So these are integrating fire networks. So all that happens with the individual point neurons is that they receive input, sum them up, and will fire under certain conditions. And a lot of the information processing actually happens in the connections in the network. So the individual, you can make very large networks of these and look at the spiking patterns and so on of the individual neurons. And if you change the property of the connectivity in the network, the synaptic properties, then you can investigate how this type of network processes information. Another approach is at the bottom here, where you have experimental details of the structure of the cells, maybe you've reconstructed the dendrites, maybe you've reconstructed the density of these cells in a given area of the brain, and you want to incorporate this type of information. Maybe you have information on the ion channels that are present on the soma and the dendrites and so on, and you can certainly build these type of networks incorporating this very great amount of detail, and then you can create these type of network models, simulate them, and relate that directly back to your experiments in your lab, as if you were sticking a probe in the corresponding section of the brain. You can run very similar experiments in these computational models. So there's different approaches to how you might model in computational neuroscience, and lots of publications in all of these different types of areas. And as I say, these are the types of models that we want to share, make more accessible on open source brain. So to give a very quick overview of just these are typical types of models that are present on open source brain. You can see on the right here, we have models from neocortex, cerebellum, hippocampus, olfactory bulb, lots of different brain regions. And they range over multiple different scales. For example, going from a single cell models to cells with less than 100 cell, network models with less than 100 cells, up to very large scale network models. And on the y-axis here, we have the models range from, as I say, point neuron, integrating fire, maybe Izakevich models, all the way up to detailed multi-compartmental neurons. So for example, this Hay et al. 2011 is a detailed layer 5 pyramidal cell model. So the framework that's there on open source brain for sharing these models, for simulating these models, covers all of these different areas. And these are the type of models we want to make more accessible for the community.
Yeah, that's great. Porrig, can you say something? So first, thanks for this introduction, because I think it was very useful, and it's quite nice to see all the different types of models that people can find on Open Source Brain. But can you comment something a little bit on the motivations behind Open Source Brain? What problems is it trying to address? How is it helping the community by providing all of these models? Is it about the improve how easy it is to share them, how easy it is to develop them, to iterate on them?
Okay, so basically all of the models that you see here have already been published. They have code for these has been released in various different formats, maybe C++, maybe Neuron, and so on. And there are resources where you can go and download these models. You can install it locally, you can install the corresponding simulator, you can run the model in the form of code that was produced by the people who wrote the paper initially. But if you want to compare one model to another, if you want to make modifications, you'll probably have to install a number of different simulators. You'll probably have to learn different programming languages for looking through these. And hopefully the code is reasonably well written. You'll be able to find where you want to change a particular parameter. And then if you change a particular parameter, you want to investigate what's happening in the network in that way. Now, what we want to do with Open Source Brain is to take a more standardized, accessible approach and try to get these models into a standardized format so that there's just one way to express these, and that's called NeuroML, and then make it easier for people without installing anything on their local machine, without installing, needing to download the code and so on, to make an easy-to-use web-based interface for actually running the models, changing some parameters without having to look at any of the code.
Okay, and can we have a look at what it looks like to work with one of these models on Open Source Brain?
Sure, okay, let me just, okay, so you can see this here. So this is one of the projects that's on Open Source Brain. So this is a project related to the Hodgkin-Huxley model. So this is quite a famous model from 50, 60 years ago, one of the first computational models in neuroscience. And on this page here, if you go to this, if you search for here, Open Source Brain and search for Hodgkin-Huxley, you'll come to this page. And all you need to do is click on the big button saying Explore Model. And what it's actually doing now is loading up Geppetto in the background. And this 3D interface, as you see here, just shows a single compartment cell. And I'm just going to show
so that
here.
blue sphere, that is the soma of the neuron?
That is the soma of the neuron. Well, it's yellow now. That's the soma. And if you click on model description, you can see more information about it. So it's a cell, Hodgkin-Huxley cell, one segment, and there's a number of ion channels present. So potassium, sodium, and so on. And on this model also, there is an input, current input. So basically all it is a single model neuron with a current injection, and it's going to spike when it receives some current injection. So what I will do is just persist the project. So this button here on the interface means that the project is saved in its current state, and I'm able to run simulations. Just to point out that I'm actually logged in on the Open Source Brain website. So if anybody does want to repeat this, you're very welcome to, but you will need an account on Open Source Brain. But once you have an account, you can run simulations and replay them in your browser. So let me just go back here and look at the list of experiments. So now that I've saved the model in its current state, I just press run active experiment, and I'll get an interface here for saying how long I want the simulation, number of processors and so on, but also which simulator I can run it on. So Neuron is the most popular neuronal simulator in the field. There's some other options. There's also options for actually running. So the default option is run in Neuron on our Open Source Brain servers. There is actually another option for running on NSG, the Neuroscience Gateway in San Diego, which provides supercomputing resources. So for larger scale models, you can actually run network models on hundreds of processors. I won't do that at the moment. I'll just run this nice simple model here.
Okay, so, and I think the drop down wasn't showing up because of the way you're sharing screen, but so essentially there was a drop down on screen with NSG and the different simulators that are available. Okay.
Yes, yes. Good. Okay. So what I've done here is press submit, and this is actually submitted to our servers, and it's currently running on the server. It's been converted from this NeuroML format into Neuron, and I'll say a little bit more about that. And when this dot turns green, it means that it's finished the simulation, it's recorded everything, and it's sent it back to the browser. So if I just do results, plot all recorded variables, what you actually see here is the membrane potential of the neuron over time. And one other thing that I can do just to animate this a little bit is apply this voltage coloring to the soma here. And I'll show the simulation time, and I can replay this simulation, getting the neuron to light up. So this is basically replaying the simulation, replaying the recorded membrane potential, getting the neuron to spike. So in this way, it kind of animates it a little bit more, what's actually been recorded, and you can see which neurons are lighting up in your simulations.
Okay, and there, Porg, when you pressed on the results button, what you clicked on was plot all recorded variables. But how can people? How do you pick which variables you would like to record?
Okay, when you press run for the first time, if you haven't selected anything to record, by default, you're given an option for would you like to record the membrane potential in all cells, or would you like to record the membrane potential in all somas? There is actually another option here that you can search through all of the variables, and so for example,
so these are all the variables that are available currently in these Hodgkin Huxley?
Yes. Okay. So if you want to record the conductance density of potassium, if you want to record the leak current, you can record all of these variables here. You can change some parameters and so on. Yes, and you have a lot of, if you click on this control panel, you have a lot of control over what you want to do.
And so what would be the way, for instance, if now you wanted to do that, say change a parameter, maybe record a couple of variables, what do we need to do? Say if someone is on open source brain now, and is it possible to add another experiment or?
Sure, I can just show you. I can copy the existing experiment here. So let me just close that. I've copied a new experiment, hasn't been run yet. So what I will do is I will record some more variables here. So I'm just going to record the current density of the sodium and potassium channels.
Okay, so you're just clicking there on the icon on the right-hand side.
Yes.
Okay.
And what I'll also do is change one of the parameters here. This one here. So instead of injecting 0.1 nanoamps, I'm going to inject 0.3 nanoamps. So the cell should spike faster. So now I've changed these values here. You can see what is being recorded. Hodgkin-Huxley population, V, Na channels, current density. And one parameter has been changed, pulse generator 1. Amplitude is 0.3, and let's run another simulation.
Okay, so for each one of these configurations, say set of variables that you've recorded and parameters that you've set, you will have one entry, a different experiment there at the bottom. And you can maintain multiple of them. That's good. Okay.
Yeah. So you can record, you can look at, so the whole idea here is that you can run multiple different experiments, you can change parameters, it's all recorded for you. You don't have to download these. You can close your browser, come back, see which experiments have been completed, and see exactly what has been going on. So let me just show you this here. So this is the these are the current densities that have been recorded. And let me just plot the membrane potential. So again, the cell has been spiking at a higher rate, and you have the membrane potential here, you have the current density for sodium and potassium, and in this way you can change different parameters. You can change the conductance density of the ion channels and perform lots of different experiments in this way.
Okay, this looks quite nice. Obviously, it's not the first time that I see it, but it's always nice to see it again. And like, I'm sure one of the things that is coming to mind is how, given that this lowers a whole lot the barriers for actually getting to run simulations, because people don't have to install anything and they can play with all of these models. So one of the things that comes to mind is how this could be at the very least, a tremendous educational resource. And are there any ways on open source brain to have some type of tutorials, say, to understand, for instance, what the parameters in a model like these are, what happens if you actually change them?
Yeah. There certainly is. Now we have a number of tutorials. We started, one of them is actually on the Hodgkin-Huxley model, and this one here is actually available from the homepage. If you click on the link for Hodgkin-Huxley tutorial, but I'll just quickly show this video that I've recorded of it. So as opposed to what I had been doing, which is going through myself, selecting things to record, trying to find some interesting variables to change. This tutorial actually has a widget or a kind of graphical screen here, which will guide you through the various steps and the various parameters you might need to change to explore the Hodgkin-Huxley model. So what you see here is just a little bit of information on the structure of the model, and you have some fields that you can change here. So the input current, sodium and potassium conductance. What I'm actually just doing is changing the input current here, hitting the button for try this model, and it does, the tutorial will do all the rest behind the scenes. It'll run the simulation, and when the simulations are run, it pops up plots of the variables that have just been recorded. So that's the current density, the conductance density here, the rate variables on the bottom middle, and on the left, the membrane potential. So all of these are popped up. If you change any parameters, like change the sodium conductance, you'll be able to run the experiment again and see the effect on the activation variables, the membrane potential, and so on. So this another example is a voltage clamp experiment. So this is another experiment that was performed in the original work of Hodgkin and Huxley. So again, you have a brief explanation of it here. You can change some parameters for the voltage clamp and just hit try. And again, the experiment is submitted to Open Source Brain. You get a brief summary on the bottom here of the parameters in your model. And again, you get plots of the current conductance densities and current densities for the model, the activation and inactivation variables. And in this way, a student can go through this, follow the tutorial, change some of these parameters, switch on and off the different conductances, and see the effect on the cell, and recreate a lot of the original experiments that Hodgkin and Huxley did with this squid giant axon to try to figure out the underlying behavior of this system.
Okay, well, this is quite nice. I'm sure there's lots of even professors that are looking at this webinar now and are thinking how this could be a great resource even to teach to their students about computational neuroscience. So I think this will spark a lot of interest. In terms of, can you say something, Porg, about where these models are coming from on Open Source Brain, and what people need to do if they have a model and they want to add it and work with it, so that they can do the same things that you're doing, but on their model, not on, say, Hodgkin-Huxley or whatever is already there?
Okay, certainly. So as I might have mentioned, the models themselves, well, one of the motivations for Open Source Brain was that we didn't want to become one central place where we store everything. We control access to who can see what and who can edit what. We really wanted to encourage people to put their models out there in as open source, as publicly available resources, and make them as freely available as possible. So while Open Source Brain is a central resource for finding these models, the models themselves are actually generally living on GitHub. So GitHub is one of the most popular code sharing repositories out there. Anyone can sign up, create an account, and start sharing their code. And the idea is that you'll share your models on resources like GitHub, and then link them to a project on Open Source Brain. And then we'll provide a way to search through the various models that are present. And if they are in a standardized format, give some extra functionality for being able to search through, being able to visualize, being able to run simulations on these. So the one requirement that we have for allowing this to happen is that if you share your model on GitHub in standardized NeuroML format, which is a standard language in computational neuroscience for expressing cell models, ion channels, networks. If you share it on GitHub in this format, linked to an open source Brain project, you get all of that functionality for visualizing the contents of the repository, for running simulations, and a lot of other functionality. And again, since the idea is that it's open source, anybody else can take a component from your model, say if there's an interesting cell, they can reuse it in theirs, and they'll get all of this extra functionality as well. So just to mention briefly that the core part of Open Source Brain providing this visualization and simulation functionality is a customized form of Geppetto. And this is what, so Geppetto is actually what interprets, reads the NeuroML, interprets that, converts it into graphical format, and handles all of the conversion of NeuroML into the simulator specific formats, running the simulations, and then making it as easy as possible for the user to share their model and give access to this for running on just inside other people's browsers. So if you do want to share your project, all you need to do is click sign up for Open Source Brain, click on add new project, and you'll get a screen like this. And all it really is give a name to your project, a unique identifier, a brief description, and point to your GitHub repository. So that's all you really need to do. And then everything I've shown there, as long as it's valid in NeuroML, everything I've shown previously is available for you and your model. You can give it a link to people to run simulations with it, visualize it, and hopefully improve it and use it for new interesting projects. Just briefly as well, if you go to neuroml.org, there's a lot of tools there to help people convert their models into NeuroML to maybe export it from one of the existing simulators. There's links to some of the recent papers and some examples and some libraries in Java and Python so that you can get started building your model in NeuroML format.
Okay, so just to recap, people create a GitHub repo. It's their own.
Mm-hmm.
They put a NeuroML model in the GitHub repo. They then go to that page you were showing a screenshot of. They create an OSB project that they put there the link of the GitHub repo that they just created, and then upon creating a new project, OSB will scan the content of the repository, will find any NeuroML models that are there, and then people will just be able to load them, visualize them, and simulate them and do the things that you were doing.
Yeah.
Okay, that sounds easy enough for people to try it. And Borg, if we could take you showed the metrics at the beginning that had a whole lot of models spanning in lots of different complexities, both in, yes, this one, going from single cells to large networks that have different levels of biological detail in them. But we just looked at Hodgkin-Huxley. Do you have some example of something. But actually, before we do that, let me ask the audience because this is a very nice picture, and I would be interested in asking people what computational models they work with in neuroscience. So there is a poll in front of you, so you should be able to pick between point neurons, detailed single cells. So for instance, if you're incorporating a detailed morphology of your model with detailed ion channels and so on, or if you're modeling more the sub-circuits type of things, or large scale networks. And I see we're at 70% so far. I will give it another 10 seconds for people to keep voting. Thank you. I see numbers going up. And it's nice to see that it's a rather even breakdown with circuits leading the way, but we have enough percentage on pretty much every category. So first is circuits, the second is detailed single cells, then other, which is a catch-all, then we go to point neurons and large-scale networks. I think this was interesting. So far, looking also at this breakdown, looking at your metrics, can we look at some other type of neurons, for instance, if there's something that is more biological details in them?
Okay, sure. I think you may have to close the poll before I can show this.
I think I closed the poll, but what I see is, okay, now I see the videos.
Okay.
I was just catching up, I think.
Okay, sure. Okay, so this is just showing an example of a group of neurons from the Allen Institute. They have recently been reconstructing sets of neurons from the mouse visual cortex, and what you see here is about 40 or so detailed reconstructed neurons from various layers of mouse visual cortex. The yellows in layer 6 up to layer 2, 3 in red. And what you can see here is that I've converted these models into a NeuroML format for visualization in open source brain. So they've released it on their Allen Institute cell types database. They can be converted into NeuroML, and these, because they're in NeuroML on this GitHub repository, they can be visualized in the browser in this way. So another set of models that have been converted into NeuroML are the cell models from the Blue Brain project. So the Blue Brain project is recreating a microcolumn from the rat somatosensory cortex, and what you see here is a layer 5 pyramidal cell that they have released. And again, this is in NeuroML format, but this example here, what I'm going to do is click on model description, as I showed for Hodgkin-Huxley, and here there's a much larger number of segments, number of segment groups, and lots more ion channels. And through this model description interface, you can click on various things here. So this, for example, highlights the cell regions. So the dendrites are in green, the soma is in blue, and the small axon is in orange. So in this way, you can also actually visualize where the active conductances are present on the dendrites and soma. So for example, here, this sodium channel, there's a high concentration on the soma. There's also present on the apical dendrites. And you can zoom in and find exactly where those are present. This one is a calcium conductance. And finally, we have IH, which is a H channel. and this is present on the apical dendrites, and it's actually non-uniform. So there's a higher concentration of this particular ion channel at the tip of the apical dendrites. So in this way, we've provided an interface where the Blue Brain project cells can be visualized in this way. You can see exactly what type of ion channels are present and where it's actually present on the cell model. One final example here is from the cerebellum. And what I have here again is a Golgi cell model from the cerebellar cortex. Again, soma in blue and the axons in orange at the bottom. And what I'm going to do in this case is show some of the properties of the list of the ion channels, but you can also show the properties of the ion channels. So this is showing the time course and steady state for a number of the activation variables in this low voltage activated potassium channel. And again, all of this information is extracted from the NeuroML description. So if you do want to look at the specific properties, compare the different ion channels that are present on the cell, you can do all of this if the model is in NeuroML format. Okay, so because there's a number of people who are interested in networks, maybe we should have a look at some networks.
Yes, that would be fantastic.
Okay, let me just close that. And can you see that again?
Yes, we can.
Okay, so this network model that I'm showing here is quite detailed. It actually consists of about 300 cells. So each one of these is an individual cell model, and this is again a model of thalamocortical network model. So each of these different colors are different layers. The red is layer 2, 3. I believe the blue is layer 4. And because there's 300 cells here, it's difficult to see the connectivity within this network. So we have a connectivity button here on the OSB interface, and this will extract some properties. There's various different ways you can visualize the connectivity. Extract some properties from the NeuroML description of the network. And in this matrix here, this is the adjacency matrix, you can actually click through it, and clicking this line along the top here highlights which one of the populations. So this is layer 2, 3, pyramidal regular spiking cell. And in this way, you can identify which the populations are, but also if you click on the individual points in this network, so each one of these colored points represents a connection between populations. So at a high level here, you can see where there's a very large group of connections between, for example, layer 2, 3 regular spiking pyramidal cells. And in this way, just kind of gives an overview of the connections that are present in this very complex network. So I'm just going to show one more here and this is also a cortical column model, but this is a point neuron model from Potchins and Diesman in 2014. This was originally developed in the NEST simulator. So you can actually visualize the connections here, and as you see in the background in this adjacency matrix, but you can also select this chord diagram representation of the connectivity. So this, again, the colors correspond to the populations in the network, and if you hover over one of these chords, you can see the projection for example, here, layer 4 excitatory cells, which other populations this group of cells is connecting to. So in this way, in this interactive widget, you can get an idea of which populations are connected to others within the network. So another option is just clicking on individual cells, and here you can see where, selecting one individual cell in this column, you'll be able to see where the connections, where it's projecting to in the rest of the column, and try to get a feel for the connectivity between different groups of cells in this network. So this is all possible as well for other types of network model. So one final example here is a hippocampal network, and this is a detailed cell model by Bezier et al. From 2016. And this actually consists of quite a large number of CA1 pyramidal cells, a number of interneurons, but there's actually 90% of these 300 odd cells here are these blue CA1 pyramidal neurons. If you click on one of these here, you can see the connections between the pyramidal cell and other cells in this network, and see where they're projecting to on the dendrites, for example. But since it's a very complex network, maybe you want to see how many other of these interneurons are present. So if you click on the control panel again here at the side, you can actually search for that cell model name. And once you find it there, you can select hide. It'll hide that population, and then all you're left with is the 10% of the interneurons in the network. So it makes it a little bit easier to see the wood from the trees. Yes, so one final connectivity widget to show you. This again is a representation of the connections in the network. All these blue dots represent the 90% of pyramidal cells. The cells at the center here represent the interneurons, and you can see there's a huge amount of connectivity between these pyramidal cells, but there's also a lot of connectivity in between these interneurons here. So in this way, you can kind of, all of these different widgets can give you a different insight into the types of connectivity that are present in these different types of network. Okay, and just to wrap up, I'll just show.
If you heard something about maybe simulations, because we were looking at the simulation of a single Hodgkin-Huxley, but now with this more complex model, we're looking more at their morphologies or the structural properties of the network. But are these type of models still something we can simulate?
Sure, yeah, okay. I'll just show again a simulation. This here is a pyramidal cell model from Smith et al. 2013, layer 2-3 pyramidal cell model. And this is obviously morphologically detailed. And what I want to do here is not just record in the soma. So I'm just going to look for everywhere that it has membrane potential. I'm going to record some different locations in the dendrites. Search for a few more. And what this cell model is receiving some background stimulation. So the dendrites are going to be active. It's going to be spiking at the soma, and we want to investigate what's happening in the dendrites when we set the simulation running. So we've selected to record some locations dispersed along the cell. There's some background input. We just hit run active experiment, set it running for a little bit longer. It gets sent to the server, and it's finished. And we just plot all recorded variables. And what you see here is a number of different traces. I think there's about 6 of these. And it's clear that a lot of these, so these are not all identical traces, so it's not the same membrane potential everywhere, but you can see here on some of these traces, there's some local spiking activity. So in this way, you can investigate whether there's activity in the dendrites, whether some background input is causing spikes in the dendrite. And obviously, if you zoom out, you can see where those might propagate to the soma, causing the whole cell to fire. But there's clearly some locations on the cell which are spiking away by themselves. Okay. And to show another example, this is from the olfactory bulb. And what this is showing is 3 mitral cells here from a network simulation in the olfactory bulb. So they're detailed, morphologically detailed cells again. And what I've done here is run a simulation and recorded every segment in the all of the cells. So what you can see here is that when the cells start to spike, it spreads out in the dendrites, reaches the soma, and causes the entire cell to spike. So on the left, obviously, is a 3D representation. On the right is some plotted traces of these mitral cells. There's some granule cells, smaller granule cells at the bottom. And in this way, you can investigate what happens in a detailed model like this. So this is just replaying the simulation at a slightly slower pace so that you can actually see where it's where the activation is going from the dendrites into the soma and causing it to spike. And you can slow it down and look at various different locations on the cell. So it just provides a way without, as I say, installing anything. You just need to select to record every location on the cell, and then you can really investigate what happens, how the cell is integrating its input, and where the spike is initiating, for example. And one final example here. So previously I had been recording the membrane potential on the cells. This case here is actually the Traub thalamocortical model. This is a layer 2-3, some pyramidal cells, a number of interneurons. And instead of the membrane potential, I've recorded the calcium concentration, the internal calcium concentration. So what you see here when the cells are lighting up is an elevation in the internal calcium. So there's background stimulation. And what you can see here is that when the cell starts to spike, there is a local elevation in the concentration of calcium. So in this way, you can also investigate this, and this is quite much closer to, so for example, population calcium imaging experiments that you might do in the lab. This is really an interface for recreating these type of experiments. You can select any different location here for recording, and then do some very close comparison to what you're actually seeing in calcium imaging in the lab.
And this looks quite impressive. Congratulations for like open source brain in general. It's like it has come a long way from 2012. I'm sure everybody agrees on that. And maybe this maybe is a good moment. Let me ask the audience, because we looked at many different things throughout these different videos, and I would be interesting to see what aspects people find more interesting or useful for their own workflows. So you should be seeing a poll on your screen, and you are able to pick between whether sharing and collaborating is the part on the development of the model, is the part that you find mostly interesting, useful, or the online visualization and simulation side of things. So essentially, without installing any tools, being able to look at your models and simulate them, or the tutorials and educational resources. So this would be something more oriented for students and for teaching computational neuroscience, the seamless access to supercomputers. So as Poreg was showing, it boils down to a drop-down, for instance, where you can decide to send your simulations, for instance, to the San Diego supercomputer, the Neuroscience Gateway Center, and whether that's something that is appealing, or if it's in general the idea of a model repository that captures all these different types of models that we've seen can range and vary across different scales. So let's close the poll. Let's have a look at the results. All right. Well, it looks like the videos were flashy enough, and a lot of people like the online visualization and simulation side of things. But second, which is, to be honest, I'm happy to see, is the portion which is about sharing and collaborating, which is something definitely that the field needs to improve, and improve also the quality of the models that are out there. Then we have, that goes hand in hand with this really, is the concept of a model repository where you can put all these different types of model. And then we have tutorials and educational resources. And last, seamless access to supercomputers that might not be as relevant depending on what type of modeling people are doing. Okay, I suppose, Poreg, we've seen a lot of things. Maybe as a last question, is there any publication for OSB that we can point people to?
Okay. Yes, there is, just recently, we've released a preprint on bioRxiv specifically about open source brains. So a lot of the examples that I've shown you now are discussed in this preprint. So there's a link at the top there, bit.ly/OSB preprint, but you can also search on bioRxiv or open source brain, and you'll be able to read about all the different functionality, how it's implemented, how we've used Geppetto under the hood, and the motivation behind open source brain, where it's at the moment.
Okay, so bit.ly/OSB preprint is the URL if people want to get into more details. Okay, so we are 9 minutes away from the end of this webinar. Maybe it is the time to go back to the audience and see what questions people have asked, and we can try to answer some of them for it.
Okay.
So let me. Okay, a question here is to start basic modeling of neural networks. What software or tool is good? What would you recommend?
Okay, it really depends on the level of detail of your model. If you do want to create some very small point neuron models with a minimal amount of code, then I'd recommend the Brian simulator. So within 10 lines of Python, you can create an integrate and fire neuron. You can create a network, a small network of these, and you can run simulations, and then it's entirely within Python. It's nice and clean, easy to install, and you can have your analysis code, your visualization code, plotting everything else in Python beside it. If you wanted a little bit more detail in your cells, if you wanted to implement ion channel models, maybe some multi-compartmental models, then certainly the neuron simulator is best in class at the moment. It does take a little bit more effort to get it installed, but once it is installed, you can certainly write in Python also. There is a lot of online documentation. Also for Brian, there's an active user community, but for Neuron, there is a book also which explains some of the models, some of the modeling concepts, but also how to build the models from scratch in Neuron. So Neuron definitely, but it will involve installing software and learning a little bit about the language itself. And also at a simpler level, Brian.
Okay, let's pick another one. If someone wants to use proprietary data to add variable to this open source model, is it something can be done on Open Source Brain as a platform, or is it a place where only public data can be used at the moment?
Well, all of the, so anything we're visualizing at the moment is being pulled from GitHub. So unless we don't have functionality at the moment, and we're not really planning on it anytime soon, to be able to pull from private repositories on GitHub, the real kind of incentive for offering this kind of visualization is that people will have to share it first, and then they can get all of this functionality behind the scenes. I mean, all of the models that you see here, they can all be downloaded locally, and you can use the tools for converting NeuroML into Neuron, for running Neuron. You can install these locally and run all of these locally and do anything you want with them locally. But if you do want this graphical functionality, then you'll need to upload it to GitHub. It is possible to install Geppetto. It is possible to install Open Source Brain locally and use it as a kind of internal web browser for your models. But for most of the work you might want to do for building and running lots of simulations in your models, all of the tool chain behind it can certainly be installed locally.
Okay, and we have time for another question at least. So how do you define the synaptic weight PORG in NeuroML?
At the moment, there is a Python library. There are Python libraries for reading and writing NeuroML. So the NeuroML that you actually see visualized there is the kind of instantiated XML, or it can also be saved to HDF5 file of the list of explicit connections in the network. So each one of those connections will have its own weight, but there are Python scripts which can write the NeuroML for you. So basically, if you want a random distribution of weights in a network, if you want them varying as a function of distance between cells, all of this is possible in Python. You just have to write your Python script using this libNeuromL Python library, and then you can give any type of weights that you want to your connections. And once you've generated an instance of that, you can put it on GitHub. You can see it, see the weights in that matrix, and obviously run simulations using those weights. But the way you'd actually put it into the network is to create a Python script which will generate the network for you.
Okay. There is, I think, maybe time for another question. So, is it possible to select the numerical method that is going to be used in the simulation?
Well, you do have some control over which simulators. So usually the simulators will decide internally their own preferred simulation method. You do have some control over, for example, the simulation time step. So if you run a simulation in Neuron, you can give a simulation time step of 0.025 milliseconds, or even smaller if you want, and it will run a simulation with that. But we don't have the very low level control over the internals of Neuron. I mean, you can certainly download your model and run it in different ways locally, but we try to keep it as simple as possible with just a single parameter for the time step for the simulation. But if you want to change to a different simulator, maybe it'll be a little bit faster. Another alternative is if you use this NetPyne library on top of Neuron. Again, it's generated automatically for you, but in that case, you can run it across multiple processors. So you can generate a network and maybe run it across 100 processors on this neuroscience gateway. So in that way, it might it should certainly speed up the simulation time.
Okay, and then there was another note, as in the metrics of models on OSB, tissue level types of models are missing, but probably the point there is just to say that it's whatever models people will contribute. So if somebody puts creates a GitHub repository and adds some different types of models that belong to some other class, they will just show up and be picked up by open source brain. So there is no bias or strictness about all of this type of model.
No, I mean, you can certainly put any type of model, any level of model into a GitHub repository. And then as far as OSB is concerned, you have all the advantages that people will know about it. People can look for hippocampal models and see your hippocampal model and download the code there. So you get a lot of those advantages of sharing and open source and all of this associated best practice, independent of the type of model. But if you do want to visualize it, the functionality we have is depending on that type of model being supported by NeuroML. So we do have quite a large scope for the types of models that are supported in NeuroML, but not very abstract or very kind of volume-based models. We are working to expand those, but first things first, we just need to have some models shared on Open Source Brain, then we can see the types of things we might need to add to NeuroML. And also there needs to be simulators out there which can support these type of things. So there is a family of simulators like Neuron and Genesis which cover very similar scopes. So we've been kind of concentrating on that superset of functionality to try to get the elements that we'll need to include in NeuroML and then support these through the visualization side of Open Source Brain.
Okay, great. Well, I think we are at the end of the webinar here. It's 4 p.m. British summertime, and for once, Britain is actually summer. So I'm looking forward to enjoy that. But I want to thank you so much for your questions and for attending the webinar today. You should get a short survey over email if you will have a few minutes. Just it will help us knowing more about who you are. And let me thank again our guest Porg here, that has done a wonderful job walking us through open source brain, which is for a tremendous resource. So I think lots of people and group will find it useful and a novel way to share and collaborate while developing computational models. So congratulations again. And as a reminder, you can follow MetaCell on Twitter. So our Twitter handle is just @MetaCell. And thanks for being with us today. And I hope to see you at our next webinar. Have a good rest of the day. Thank you. Thanks, Porg.
Thanks.
See you soon. Thank you. Bye-bye.